Hi everyone, my name is Miao Xu. So today my first topic is the introduction of platelet and its role in thrombus formation. First of all, what are platelets? We know they are the smallest blood cells in our circulation. Their color is white, and interestingly, they have no nucleus. They're basically generated from the cytoplasm of megakaryocytes. With electron microscopy, we now can have a better look at the platelets. Resting platelets are round and discoid. When stimulated with agonist, they can either become spider-shaped or fried egg-shaped. So how is platelet identified? A number of different scientists would recognize as the first to describe platelets. Oshler clearly described platelets in his papers both in 1873 and 1874. He recognized their disc-like structure, and they can rapidly form aggregates when removed from the vessel. But he was not sure whether platelets were normal cells or exogenous organisms, such as bacteria. 7 years later, another scientist named Bizarro used elegant and compelling studies to identify platelet, but also find out their role in hemostasis and thrombosis using intravitreal microscopy in guinea pigs. Studies in recent years revealed the versatility and complications of platelets. They involve in many other biological processes, such as immune responses, angiogenesis, tumorigenesis, metastasis, and liver regeneration. But of course, they are essential for the hemostasis and thrombosis. In a bigger picture, how is platelet plaque formed? Endothelial cells form the inner layer of blood vessels and preserve vascular homeostasis by preventing adhesion of circulating platelets. However, as signs of vascular injury, endothelial cells are altered or extracellular matrix is exposed. This process, aimed at arresting hemorrhage in case of bleeding, may in pathological conditions cause thrombotic vascular occlusion. We will focus on the mechanism leading to the initial interaction of platelets with thrombogenic surfaces and the subsequent adhesion and activation. The platelet adhesion, activation, and aggregation all occur in the blood vessel and are influenced by blood flow. The blood flow in the vessel is characterized by a velocity gradient from maximal at the vessel center to minimal at the wall. This is caused by a shearing effect between adjacent layers of fluid that is greatest at the vessel wall and minimal at the center, shown as the black arrows in the picture. Platelets have molecules on their surface to sense the shear stress in the blood flow, and when the endothelial cells turn off from non-reactive to reactive, it will cause a disturbance in shear rate. The change of shear stress can lead to platelet adhesion. Several extracellular matrix components interact with platelet, such as collagens, von Willebrand factor, that is VWF, fibronectin, laminin, thrombus pointin 1, and so on. Collagen and VWF are well characterized in platelet adhesion. Today, we will discuss VWF. VWF supports platelet adhesion and high shear stress through binding of its A1 domain to glycoprotein 1b alpha in the GP1b9-5 complex, shown in the left picture. The picture in the right showed a representative binding mode between VWF A1 domain and the N-terminal of GP1b alpha in green color. Platelets adhere to VWF in a two-stage process. Initial tethering is mediated by GP1b alpha VWF A1 domain bonds that form rapidly but have limited time, that thus cannot support the stable adhesion. Therefore, the platelet will roll along the adhesive surface slowly. Because physiologically, platelets and VWF are both present in the blood circulation, so the unstable adhesion can prevent intravascular platelet aggregation. But if there are new bonds formed, the platelet will remain in contact with the substrate while translocating at low velocity, and followed by platelet activation and formation of stable bonds mediated by integrin alpha-2b beta-3. The binding of VWF to GP1b alpha induces pulses of increased cytoplasma calcium concentration. Outside-in signaling events following the binding of GP1b 9-5 to immobilize VWF trigger an initial response leading to low-level integrin alpha-2b beta-3 activation, which is sufficient for bonding to VWF and stable adhesion formation, involve transmembrane calcium flexes, and then lead to full alpha-2b beta-3 activation. As shown in picture A and B, the picture C here depicts stable adhesion and requires additional adhesive interaction, including GP6 engagement of fibrillar collagen, the ligation of multiple beta-1 integrins by their responsive ligands exposed in the subendothelium, alpha-2b beta-1 to collagen, alpha-6 beta-1 to laminin, alpha-5 beta-1 to fibronectin, as well as the binding of integrin alpha-2b beta-3 to VWF. The binding of integrin alpha-2b beta-3 to subendothelial VWF secures stable platelet adhesion to the vessel wall and is binding to fibrinogen facilitates platelet aggregation. Intracellular calcium mobilization, platelets undergo shape change and granule secretion that coincide with the formation of stable platelet aggregate, which are interaction between platelet and platelet, ADP, TXA2, and thrombin bind to their respective G protein coupled receptor to amplify platelet activation. These signaling events combine to maintain integrin alpha-2b beta-3 in a high affinity state in which it can bind to fibrinogen and VWF to enhance adhesive strength and platelet cohesion and then promote thrombus propagation. Thank you very much.